So last video we finished off by showing a plan diagram of the leaf lamina. This is, of the leaf midrib. This is a cross section or a transverse section or a leaf lamina section. So we're not going to draw a plan diagram of this right yet, but we have here the upper epidermis. We have our palisade mesophyll, these vertical cells with nuclei and chloroplasts within them. We also have our spongy parenchyma down here. And the spongy parenchyma down here, we also have air spaces mixed within. Now, we also have the lower epidermis. This lower epidermis will periodically be punctuated by a stoma. A stoma, or stomata plural, is the opening in the leaf epidermis that allows for gas exchange in the plant leaf. Now, gas exchange in the plant leaf is for the diffusion of carbon dioxide and oxygen. It is not for the diffusion of water vapor, as we'll discuss later. It is for carbon dioxide and oxygen exchange, specifically for carbon dioxide exchange. So this stoma, is flanked on either side by guard cells. Guard cells are the openings to stomata. Now here we have these vascular bundles surrounded by bundle sheath cells. Those bundle sheath cells are parenchymatous cells that surround and sheathe a vascular bundle, thus bundle sheath cells. So in a vascular bundle, we will have xylem facing the upper epidermis, and we will have phloem facing the lower epidermis. Xylem being a tissue with thickened cell walls and large lumen, an empty intracellular space, and phloem having two different cell types within, being a compound tissue, having small nucleated cells and larger phloem sieve tube elements that are paler in appearance. So here we see a bundle sheath cell that has been caught edge forward. Effectively, we are catching a tube that has been cut as if you took a hot dog and you cut it into little coin-shaped slices. Whereas here we are seeing the lengthwise section. So we're seeing a longitudinal section of a vascular bundle here, and we're seeing a transverse section of a vascular bundle here. So we're seeing the two different angles of cuts for these bundle sheath cells. So note what you're looking at. You're not looking at some kind of worm thing. You're looking at vascular bundles being cut in two different directions. Now, this is a drawing of that vascular bundle. We also see a cuticle above here as well. We'll see that typically on the upper surface of a leaf, sometimes on the lower surface as well. A cuticle being a waxy layer that's designed to prevent water loss in the leaf. Now, let's talk about roots for a moment. Roots will have a large exterior with a very thin epidermis. Now, if we were to catch this root in the root hair region, we might see periodically a root hair here or a root hair there. But that's not shown here. We are not lucky enough to have caught one of those. Now, this epidermis is just a mere thin layer right over here, this edge of cells. These cells, this much thicker region, is going to be our cortex. Now I'm going to enlarge this center region so that it's easier to see for the camera. And that central region is going to be our vascular cylinder. Our vascular cylinder consists of a number of different things. Now here's a close-up of a different vascular cylinder, but we might as well call this the same one. Now our vascular cylinder is composed of our endodermis, our pericycle, our xylem, and our phloem. So these different tissues right here, we are noting that there is a distinctive layer. The stele, the S-T-E-L-E, and I'm not sure I'm pronouncing that correctly because, frankly, I've never actually seen it pronounced. I've only read the word. Never heard it pronounced. I've only seen the word written. Is the area within the endodermis. It is the pericycle, xylem, and phloem. Our vascular bundle is our phloem and our xylem tissue. We can see our xylem tissue are larger cells with thick cell walls, and our phloem tissue are our compound tissues with large cells and smaller nucleated cells, metabolically active as well. Our endodermis cells are out here. They have periodically thickened walls as well, and they are distinctly different from our cortical cells. Our pericycle is a layer of cells that is not quite phloem and certainly not xylem. Now, here is a drawing that is almost a Cambridge-approved plan diagram, but not quite. And this is giving us our subrated, this is showing us our subrated endodermis with showing us individual cells, which is why this is not an approved Cambridge plan diagram. It's also labeling inside the plan diagram, so that's not approved. But it is showing us individual root hairs. But it's showing them as cells, which is not permitted. Now, this is a longitudinal section. This is a longitudinal section of the root hair region, the root tip. Down here we have our root cap area. We can see in our drawing of this root cap the different kinds of cells. We can actually see the difference between these cell types. We can see this meristem region right here, this denser cluster of smaller cells right around here. We can also see our zone of elongation. This is a zone where the cells themselves will elongate as they divide and differentiate from the meristem region. And we can also see in our image of the picture, the drawing, our zone of differentiation, where our different cells will turn into our different types of tissues. Now, it's very difficult to find an image of a root tip that contains both the root cap, the meristem, the zone of elongation, and the zone of differentiation in one image at appropriate magnification, because under magnification, this is a very large structure. Without magnification, it is really difficult to see these structures at all. Now, these different zones right in here, we can definitely see that there is a difference, a distinct difference in the tissues of our epidermis, our outermost layer of tissues, and our cortex tissue, this lighter colored tissue right over here. And definitely our vascular tissue, or our soon to become vascular tissue right in here. Now, let's talk about the stem for a moment. We'll talk about more monocots than we'll talk about more dicots than monocots, but for the moment, let's compare them. In a monocot stem, we will have our vascular bundle arranged in a complex pattern, in a somewhat seemingly randomized pattern. Whereas, and mixed in here is going to be ground tissue. Whereas in the vascular bundle, in the dicot stem, we're going to have a much more organized, orderly arrangement here. We're going to have our epidermis around here. We're going to have our cortex in that inner layer. We're going to have vascular bundles with sclerenchyma right here. We're going to have these medullary rays in between these vascular bundles. And we're going to have the pith inside. So I'm going to draw a simplified version of this because I don't want to draw that many vascular bundles. Or rather, how about this? Let me just draw a wedge of this. I'm just going to draw the upper... That is not a good pen. I'm just going to draw the upper wedge of this. You know what? Let's make that larger for the camera. I'm going to draw the upper wedge of this. Alright, so I'm going to draw this layer of epidermis right here. I'm going to draw this layer of cortex right here, considering that cortex is particularly thin. I'm going to draw considerably larger larger bundle structures. Go. And we're going to have some tissues in here. That's going to be the sclerenchyma. So that's going to be our sclerenchyma. That's going to have an attachment there, there, and there. That's what we're going to look like with that stuff. We're going to have our phloem on the outer edge, and then we're going to have our xylem tissue on the inner side. Okay. And then we're going to have pith on the inside here. So these being our medullary rays. That looks about right. There's actually yeah. So this distinctive tissue, there's a little bit of something in there. So we're going to put just a little edge right here. That's some parenchymatous tissue right there. So we're going to put a little edge of parenchyma in there. And so we're going to label these xylem, phloem, sclerenchyma, pith, yeah, my cortex is another layer for some reason. I screwed this up. Let's erase that line since I can. Cortex. And my epidermis will be a little bit thick for my liking. I could have made that thinner. Probably should have made that thinner. Epidermis. And this will be down here. Parenchyma. These in here will be a medullary ray. All right? So a medullary ray is a ray of ground tissue from the pith to the cortex, this stuff right here. So our sclerenchyma is not part of our vascular bundle. It is a part of the tissue that is structural and supportive, but not ultimately part of the vasculature. It does not conduct fluids and nutrients and what have you. So now we've labeled everything. Now, here we have a plan diagram that's slightly less weird looking than mine. So we have our medullary ray, we have our endodermis, we have our cortex. Oh, this one shows an endodermis, which is not actually shown. There isn't one here, but okay. Now, endodermis may or may not be present. It's unlikely for you to find one in a Cambridge question. So we would just remove that endodermis altogether. Cortex, epidermis. If you find a hair in the stem, they are not root hairs because this is not the root. They are going to be epidermal hairs. So we have also sclerenchyma, phloem, cambium, and xylem here. And our pith. Our pith is a parenchyma as well. Now, this is a longitudinal section. And no, your video quality did not just suddenly go down to 480p. It is actually graining on the slide itself. Sorry about that. So, if we remember, we have our epidermis on our outermost layer. Our epidermis on our outermost layer. We have our cortex in our next outermost layer. Oops. Our cortex in our next outermost layer. So that's going to be our next outermost layer. We have our vascular tissues next. We have our vascular tissues next. So this is going to match up in order with our tissues that we saw in our transverse section. So the order of tissues in a longitudinal section is going to match up with the order from outside to inside. Beyond the vascular tissues is going to be pith. And then beyond the pith is going to be more vascular. And then beyond that is more ground. And then beyond that is more dermal. And then air again. So the difference between a monocot and a dicot, just broad strokes overview. Monocots are embryos with a single cotyledon, pollen with a single furrow or pore, flower parts in multiples of 3, major leaf veins in parallel, stems and vascular bundles are scattered, and the roots are adventitious and secondary growth is absent. In a dicot, the embryo has 2 cotyledons, seed leaves. The pollen has 3 furrows or pores, flower parts are multiples of 4 or 5, and the major leaf veins are reticulated. The stem vascular bundles are in a ring pattern, and the roots develop from a radical, and the secondary growth is often present. Now, don't get cute on me and ask, well, what about 15? That's 3 times 5. No, don't get cute. Not funny. All right. So, because honestly, how many flowers do you find with 15 petals? Yeah. So, this is a picture from the textbook.